Hello everyone and welcome to Chapter 18 Part 2B Estimating Species Abundance. Now there's actually three parts, three questions here that we answer in Part 2 biodiversity. And number one is how to calculate the biodiversity of a habitat, and we have seen how this happens using Simpson's Index of Diversity in the previous video. In this video, we are going to answer this question instead: how to assess the distribution and abundance of organisms in a local area. Okay, so the first question is, what is the difference between abundance and distribution? When we talk about abundance, we are really talking about number. So how many organisms of this species are there. And when we talk about distribution, we are talking about really how it's arranged in the particular area. Is it really, if there is a sea nearby, is it near the beach or is it further away from the beach? Like, how is it distributed? How is it arranged in that particular area? So that two terms here is a little bit different. Now, to assess this two things, we can use three methods, and these three methods work slightly differently. Okay, first of all is random sampling using frame quadrats, systematic sampling using line transects and belt transects. These are actually two different methods, and the mark, release, and recapture method. You realize here that they are all paper 5 related, and that's because in this particular video we're going to learn a lot of methods. Okay, and they can ask this in paper 5 when they ask you to design your own experiment. So let's start by differentiating these three methods. Number one is random sampling using frame quadrats, and this is usually for small plants or small slow-moving animals. A frame quadrat is a square thing, a square frame, usually made out of wood or some metal, and within that big square, usually 1 meter times 1 meter, it's divided into smaller squares. So you can imagine why this is used for small, slow-moving animals, because if you want to count the animals, you want it to be small enough to fit into the 1 meter times 1 meter frame, and also you don't want them to move that fast because you want to count them. Or alternatively, you can be counting plants which don't move anyway, but it has to be quite small as well, fitting within the 1 times 1 meter quadrat. Okay, just so you know, you cannot misspell this word. It is not quadrant, it is quadrat. It is not, I don't know how you can spell this, quarter or some sort. It's none of these. This is a reject. It has to be spelled quadrat. Okay, so pay attention to the spelling here. Alright, the next method we're going to be learning is method 2, which is systematic sampling along line transects or belt transects. Now, this is also for plants or small, slow-moving animals, and in the difference to random sampling using frame quadrats is that it is actually used to find the distribution across an area. So random sampling and systematic sampling really differs here as well. Random sampling is like you take the quadrat and you randomly put it in a spot in the habitat. However, in systematic sampling, you are actually sampling, so taking different samples or using the quadrat along a line, so it's not random. Okay, or we can use a belt. We will go into the details later on. Method number 3 is mark, release, recapture, and it is for mobile animals, usually smaller animals, not too big, right? And this is really used to estimate the numbers of these animals, because we can't measure them using quadrats especially, and there are too many of them to count anyway. So what the idea is to mark them on the first day when you capture them, then release them into the wild, and then recapture a second batch and calculate the species abundance from there. We will get to the details later. For now, let's go to method 1, which is random sampling using frame quadrats. Let's go through the procedure first, and later I'll show you a little video of how it's actually done in the field again. Okay, because we don't get to go out, but people in the past before lockdown have went out there and done a YouTube video on it, so why not? Anyways, let's just dive right into this. Okay, so what's the first thing you should do when you come to an area where you want to do random sampling is you need to define the area, to fence up the area to know where you're measuring. So you mark out the area using to be sampled using a string and marker pole. In this case, they use some sort of tape. That's fine too. Okay, you mark you usually use the poles at the corners, and then you basically mark the area out with string or tape. And if you use string, you probably will mark it at every 1 meter, okay, so that you know at which point the measurements is, okay, you know you know how big the area is. And if you use a tape, then that's pretty much very convenient, okay, because you can use the tape to act as some sort of x and y axis so that you can randomly generate a number to obtain coordinates. Okay, so in this case, you can see here that the coordinate is 8, 10, and you can use a random number generator app on the phone in order to do that. Now you can find it on Play Store or Apple. I can't believe I'm actually promoting this, but if you Google it or whatever, you will find it. And you can see here that you can actually set what number you want to start it from to what number, and then you can randomly generate a number here. Okay, and then it will give you a random number for the y-axis, and you can do it again for the x-axis. Just change the range according to what you need. Yeah, so that's a number generator. Okay, then you basically put the quadrat spelling is important, put it on the ground to obtain those random coordinates to obtain species abundance, obviously. The quadrat is usually 1 times 1 meter, as I said just now, and the squares in between, in the middle here, is usually 10 cm times 10 cm. Each square is around this measurement, but this could differ. Okay, this is the standard one, but it could differ based on the question, so make sure you read the question properly. If not, you can tell them that you are using this quadrat size. This is the standard one. Alright, once you put it in the floor, of course you need to identify the species inside, and obviously you cannot know the species of this grass, for example, other than using some sort of picture or googling it or using some nature guide. This is the mark points when it comes to paper 5. A key is basically like a picture with some description. A nature guide is a book where you can actually buy nature guides at bookstores, and basically they have a catalog of different species, descriptions, and uses of that particular plant or species. So yeah, you can identify species using that, and then you can count them. You can estimate the species abundance. Now there are 3 ways to do so. The most basic and the most common is to count the number of individuals per unit area to calculate species density in that particular quadrat. So in this case, we can see that quadrat measurements are different. There are only 5 times 5 boxes here, and this diagram tells us that it's actually 0.5 meters times 0.5 meters. It's always going to be a square, yeah? And in each individual box is 10 cm times 10 cm. So we can use this. We can basically look at this diagram and count. This is quite straightforward. So we can see there are 3 flowers here in this particular quadrat. And to find the species density per unit area, we need to divide it by area, not distance, not length, okay, area. So it's 0.5 times 0.5. And you realize here that we have not counted the flowers that are actually at the boundary and are cut into half. We only count the ones that are fully in the quadrat. That is one rule that we will always abide by. So if you divide it, you should get something like this. Okay, so 3 divided by 0.25 meters square, which is 12 meter per meter power of -2. So this is this species density is 12 per meter square. How about here? We can do one more example here. In this case, we'll count the number of daisies. This daisy seems to be slightly on the boundary. I don't think we'll count it, I guess. That's sort of subjective as well, right? A bit annoying. But yeah, we're not going to count that. We're just going to count 1, 2, 3, 4, 5 daisies. Okay. 5 daisies, and again, to calculate species density, we're gonna divide it by 0.25 meters square, okay, which is again the area, okay, and you calculate 0.25 meters square by multiplying 0.5 to 0.5, and if you do this, you'll get 20 meters square. 20 per meter square. So this is the species of density of daisies, these are daisies, in this quadrant. So this is actually the method of counting individuals per unit area, aka species density, which is just one method to estimate species abundance. You can see that it's actually quite straightforward when it comes to little flowers like this that stand out from the green background. But it's kind of hard when it comes to things like this. So you can see this grass is kind of patchy, and you can't tell if you can't tell how many individuals there are. These individuals are not isolated, they are small and they are overlapping. Okay, in this case as well, I can see this quadrant here, it's kind of overlapping. So yeah, in this case, we can calculate species abundance using two other methods. One is that we can estimate percentage cover. In this case, we have 25 boxes, so I assume it's 0.25 meter times 0.9 meter, but because it's percentage, it doesn't really matter, really, because we are looking at 1, 2, 3, 4, 5, 6, 7, 8, 9 boxes out of 25, so 5 times 5 is 25, right? And then we are trying to calculate a percentage, and that should be around 36%. Yes, yeah, 36%, which gives us the percentage cover. Now, alternatively, you can calculate it this way. Obviously, this way is much more straightforward. Okay, this is percentage cover. Now, what if percentage cover is it's like this one is just ngam one box, right? It's just nice in that box. What if it's kind of like the grass just now? It's kind of patchy, and it's quite hard to calculate the percentage cover. So in this case, you're gonna have to estimate the percentage cover instead, and instead of saying that, oh, it's about, say, 20 to 30% coverage, you use some sort of abundance scale. And abundance scale, there are two here, Egg4 and brown blanket. In the exam, you do need to name it, so you have to name either this or this. It will come out in mark schemes, yeah? So what is the Egg4 scale? Egg4 scale stands for abundant, common, frequent, occasional, and rare. And basically, each of these categories have a range of percentage cover. So instead of saying that it's about 30 to 40 or 20 to 30%, instead of saying that, you can say that, oh, according to the Egg4 scale, it is an occasional species. The species is put into the occasional category. Now, in contrast to the Egg4 scale, the brown blanket cover abundance scale, I don't know how to pronounce it. Braun blanquet. Who knows? But anyway, the Braun blanquet cover abundance scale uses a numerical score instead of Egg4. So it has 5, 4, 3, 2, 1 and plus. So sometimes they have it in R as well, plus or R. Depends. R stands for rare, plants. Plus is also actually very rare. And again, each of this score has a range of cover. So again, instead of saying, oh, the coverage is around 20 to 30%, you say that, oh, according to Brown Bunkert's score, it is around the category of, say, 3. It's a Brown Bunkert score of 3. So yeah, that's how you use the abundance scale. And again, percentage cover and abundance scale are both for individuals that are very hard to isolate, very small species of plants, that it's just very difficult to isolate and calculate individuals. If this is not the case, then we should always count the number of individuals, and this is the more common way of doing things. Now, of course, all we've talked about today, just now, is basically just one quadrat, and what you want to do is actually repeat that many times. So this one is like just a little grid. This is actually the habitat, and this is actually many quadrats in there, and the more quadrats you have, the more reliable it's going to be. So of course the next step is to repeat it many times, not just 3 times. Don't be crazy, don't be ridiculous. 3 times is too little. If you have like a habitat that is 10 times 10 meters, you're gonna repeat it many times. Even repeat sampling at different times of the year or seasons to make sure you have reliable results, and then obtain a mean abundance, so mean species density or mean percentage cover, etc. Now, of course, when it comes to paper 5 questions, other than repeating, there's also a mark for safety. And you would think that it's a low-risk experiment, right? Because you're just going to the field with a frame and a few tape measures and poles. But in nature, things can get, as you know, in Malaysia, if you go out to the field, there's a risk of getting bitten by insects. There's allergy. There might be allergy reactions to pollen, especially angmos, allergy reaction to plants, and you gotta have precautions. So wear masks, wear gloves, and protective clothing, etc. You know the drill. Okay, now that we have done the general concept, let me show you a video, and I'm gonna voice over the video so that you get an idea of how it really looks like in the field. Again, we cannot go out of our houses right now, and I cannot demonstrate it with you right now. I would love to do so, especially in our own MCCAL field, but at the moment we can't. So let's look at someone else do it. So this here is someone else's experiment, and they're basically investigating the effects of a name environmental factor on the distribution of a given species. Specifically, they are actually looking at investigating the effects of trampling on the abundance of plantigo species on grassland. So trampling is basically whether the area is walked on or not, and basically they will have 2 areas. So 2 areas, I thought it might be easier if I draw it for you. 2 areas, one with trampling, one without trampling, and then what they're going to do is they're going to use a frame quadrat, and they're going to randomly sample the grassland and see how many species of the plantigo species plant that they can find. So you expect that the area with no trampling would probably have a higher abundance in that species of plant. Anyways, after that, they actually showed us in this video what are these 2 different plants we're looking for. So the plantigo species has these 2 plants here, ripwood plantain and the common plantain. This is not bananas, by the way. This is just a type of grass. Just happens to have the same name. And basically, you can see how they are different, and how they are different from normal grass as well. They look quite unique. Okay, this is them telling us that, hey, this is how the leaf looks like, although it's a bit teared here. There's a thin stem, and then it slowly lobes. It has a very lobed leaf. It's quite round. Why did they give us such a sport leaf? Okay, and then this ripwood plantain has quite a large leaf, quite long, and it has like little flowers here that are white, but not necessarily, you know, flowers are not necessarily always present. Okay, and the first thing they did was actually to see, they actually marked out the area that they want to, and then they're using tape, and this is what they are showing you. This tape here, you can see there are actually little markings here because they actually took a piece of string. Sorry, this is not tape, this is string, and they actually marked out every 1 meter so that they know exactly where to put the quadrat later. Okay, and they use this, yeah, they're using random sampling to really avoid bias and provide a representative result of the area. Okay, so this is the quadrat that they're using here, and it's made out of metal. It can be made out of many materials, obviously. This looks quite factory made. I wish I had one. We had to make our own one, you know, for MCKL labs. Anyways, yeah, so you see that they will put this on the ground as such. Can I enlarge this? No, it's a bit nuts. But yeah, you can see that they are actually looking at it, and you can vaguely see some plantain species. Alright, whoops. Go away, results table. You can roughly see some plantain species here. You can see these ones are quite low. It does take some observational skills to be a ecologist, a scientist involved in ecology, basically. But yeah, you can see that, and you can count that, and you can actually put that in a table. So they do this multiple times. I think they're doing it 10 times for trampled and 10 times for the untrampled area. Okay. And yeah, each of these particular quadrats are randomly placed, and they're using the random number generator to do so. And yeah, they fill in this table, and what you're going to do is after that you are going to calculate the mean frequency of the plantain in each area. Okay, you can do species density, but in this case they're doing frequency, and calculate the standard deviation for each area and see whether the standard deviations overlap. Alternatively, you can also try and see whether the standard errors overlap. So by doing this, you're basically looking at 2 sets of data found in 2 areas. You're comparing that, and you can use a statistics test to figure out whether it's different, right? You can. And the way to do that is to use a t-test. So yeah, they do use a t-test. Whoops. To calculate the mean values of 2 sets of data and see whether it occurred by chance or they are actually significantly different. Ta-da! And that's pretty much it. I will link this video with its British accents and its actual sort of descriptions in the description box below. But yeah, that's pretty much how it's done. Moving on to method 2, which is systematic sampling along line transects or belt transects. This again is 2 different methods but very similar. Systematic sampling is really not random. So just now you're using a random number generator. So as you've seen in the video, it is to really avoid bias and have a representative sample. But in this case, we are really using a line and putting those quadrats along the line. In the case of belt transects, or just measuring sampling along a line so that we can find the distribution of plants. So we do that on purpose, not because it's biased, but because we want to see how communities or distribution of different species changes across an area. So for example here, this sampling, systematic sampling, is used from grassland into woodland. So they pull like a little tape across, and basically they are taking samples from the grassland to the woodland and seeing maybe how this species change. Maybe there's flowers in the grassland but not near the woodland, and how to figure that out, you count using this sampling technique, this systematic sampling technique. In this case here, you can see that they are actually taking measurements across a river bank of some type. I assume that this is a river. I don't actually know. It's not very obvious here. I'm just guessing. So they are taking it across the river bank and seeing if the distribution of species changes as well, maybe nearer to the river bank it would be have more seaweed, and then maybe further away there's less seaweed, okay, and so on and so forth. So this is really about distribution, okay. Systematic sampling is to really find out distribution across a specific area that changes. And the idea is that we are measuring species abundance at set distances along a transect or line. Okay, it could be line transect or belt transect. So what is the difference here? So line transect is generally recording what is touching the line or string. So this is a line transect here. So you pull a line across the area, and whatever that touches the string or whatever you see that is nearby the string, you will calculate it, you record it. Whereas belt transect is slightly different. Belt transect actually uses quadrats, but unlike random sampling, where you put it randomly, okay, in systematic sampling or using belt transects, you have a line across as well, and then you use quadrats across those lines. Now those quadrat can be like end to end like this, and this is called a continuous belt transect. Basically, you have one quadrat here, then you turn it over, so you flip it so that it's end to end like this. And you do this multiple times, keep flipping it, excuse me, and yeah, this is continuous. Or you can actually use an interrupted belt transect, so use regular intervals. This one is 1 meter apart, okay. So you, instead of, yeah, do this instead of continuous, because obviously this is much more work than this one is. Interrupted belt transect is much less work, but obviously continuous belt transect will give you a more accurate reading. Anyways, so this is the two types of transects. The procedure is kind of the same, mostly the same. I'll tell you where it differs. Okay, first of all, in both line and belt transects, you are laying out a line or a tape or a string across a certain area. You measure the area using measuring tape, minimum around 10 meters, because you want it to be long enough for at least a few quadrats, or yeah, you just want it to be long enough to have a reliable measure. In the case of line transect, you calculate the species that touches the line. In case of continuous, of course, it's turning the quadrat over end to end. Interrupted, you place it along the line in regular intervals. For all this for this two, of course, you will use the same quadrat size throughout. This is the constant variable here. Okay, then in the quadrat or at the line, you use a key or nature guide to identify different species, and you record those species down at each interval. Then you use these ways to estimate species abundance. Obviously, this only applies for belt transects, okay? Because in the quadrat, this is how you do it. You could either find species density, count the number of individuals or frequency of individuals, estimate percentage cover, or you can use some sort of abundance scale. And of course, because you only did one line, right, and a few quadrats, that's not enough, and you gotta repeat that sampling. For example, this is your first line, maybe this replicate one. This is your first line here, and you've done one line. You want to make sure that your distribution is reliable, so you do it another line along the same area. So this one, they have a 5 meter gap and did another one, and they have a 5 meter gap to the right and did another one, and see, so that they can see how those how the distribution changes, not only in one line but in a couple of lines, so it's more reliable lah. Okay, and of course you do this at different times of years and seasons to make sure that your results are actually reliable. When there's repeat in a paper 5 question, there's always safety, and the safety measures here are the same as before. If you're allergic to pollen, wear a mask. Allergic to plants, wear gloves to protect growing. Even if you're not, wear them, because safety first. Ta-da! So yeah, that's how you do belt transects and line transects. I feel it's kind of intuitive, okay? I think kind of makes sense too. But let's look at a video so that we can like have a visual of how it really works. So this is a video of how to conduct line sampling. I think they're either Indonesians or Malaysians, I'm not sure, but they're very cute. They're extremely cute. Anyways, I'll let you watch a little clip and I'll take over from there.
For us to begin, we will need the following materials. A calibrated transect line. It may be 10, 25, 50, or 100 meters long, with longer transects used for more widely spaced organisms. But today, we will only use 10 meters. Just for the sake of demonstration, we will also use wooden pegs. These will serve as posts for our transect line. And now, for the sampling proper, first, we need to randomly select a sampling area. Come and join us.
Okay, I really don't know what they're doing, but basically they want to close their eyes and then they point around and then they want to like randomly select an area to do so. That's kind of unnecessary because the whole point of line transect sampling is really to find how the distribution of organisms is across a particular area. So you should already know the start point and end point. It's just like where on the shoreline you want to start. You do want it to be away from human beings so that they won't trample whatever species you want to measure. So maybe just give them the benefit of the doubt. Maybe that's what they're doing here. Okay, anyways, I'm just gonna skip to what's important here. Basically they kind of found a place, I think, eventually. Did they find it? Oh no, they go on for a while. Okay, yeah, so here they found it and they are gonna set up their line transect. You can see they're using this some sort of tape. I think that is a tape. They tie it around a plant or maybe a pole that they have just now, and they stretch that line across the area. They fast forward that video themselves. This is normal speed, and what they're gonna do is they're gonna count it. So let's watch them do it. Hang on a sec. Here you go.
Either separated or overlapped. Thank you.
Okay, sorry for cutting you off there, dude. But as you can see just now, I will just backtrack a little bit. They are literally counting like what species touches the line or what species is hanging out around the line, and they're calculating how many types of this same plant, this same species of plant can be found along this line transect. As the person said, this line, this tape measure they're using here is really around 10 meters long, but you could use this more tapes that are 25, 50, 40, 50 to 100 meters long, and yeah, people actually do that in real life. But you can see a problem here, which is kind of dumb, because what if the species is like right at the side of this line right here? Okay, how about this species right here? It's not touching a line, but it's there, right? So why aren't they taken into account? So I guess that is a weakness of the line transect. It might not be very representative. It's easy. Okay, it's easier to do than a belt transect, but it's not taking into account an area, it's just taking into account that particular line. So let's go and see what a belt transect will look like. Okay, so this is an experiment to do with belt transects. If you look at the original video that I will also link down the description box below, you realize that they called it a line transect, but it's not actually a line transect, it is a belt transect. The video got it wrong. But it is a pretty good video on how the experiment is done. So let's just watch it anyway, even though one part is wrong. So basically, they are trying to investigate interspecific competition by trees on the distribution of white clover. And clover is exactly what you imagine it to be. It is this like you know, the four-leaf clover that represents good luck. Usually it has three leaves, and actually it's quite a small plant, as you can see, relative to the size of the fingers right here. And yeah, you are gonna measure it and see how the distribution differs from a tree. So maybe nearer to a tree there'll be less clovers, and further away from a tree then there'll be more clovers. Okay, that's the hypothesis here. So what they are doing is they're showing you the line that they're using. You can see that there are several markings on the line here. You can see here and here and here, and that's actually a marking every meter to tell you how long the string is. So I think they did it for 10 meters if I'm not wrong. Okay, that's the marking right there, and they basically tied it to a tree. There you go, tied a tree and stretch it out along this area. Okay, and whoa, shaky camera, and you can see the end of the string right there. And they're gonna use a quadrat, as you can see in the corner here, in order to carry out the experiment. Okay, so essentially they are taking the quadrat, and where was that? Whoop. Quadrat, okay, is positioned along the line. This is not called line transect. Again, this is called a belt transect. Moving forward, okay, and you can see now the clovers in the quadrat placed right beside the line here. Now, there is a problem. You see, the clovers are actually extremely tiny. I'll go back a little bit. It is really tiny. See them? This is clover. This is a clover. They are very, very tiny, and as you can see, there's many grass covering it, and they are overlapping. So it's kind of hard to calculate individuals, but it's very, very possible to estimate percentage cover. So you can do this in two methods. You can just estimate a percentage, you know, or you can use an abundance scale, like the Egg4 scale or the Brown-Blanque, I don't know how to pronounce it, scale. And yeah, you can use that. In this case, they actually estimated the percentage cover. They show, she's just describing that to you right now, and they actually show you the results table in a moment. Tiny clover leaves, yes, we get it. Oh, there you go. So they show you, this is from tree. Oh, it's 20 meters, not 10 meters, I'm sorry. And they're just estimating, like just eyeballing it. And I think the video tells you that they actually counted the squares which have clover and considered that as, and used that to estimate the abundance of clover. Okay, fine, sure, you can do that as well. And yeah, that's, and then fill up the result table. Now they also have this extra tool here, which is quite peculiar. This is not a quadrat. This is some sort of way to sample by point. So instead of the quadrat, this is an alternative tool that is not covered in your syllabus. But I'll tell you what it does. What it does is that you put this into the ground, you stab it into the ground, you lower the pins, and whatever, if there's any clover along this line, it is counted as a frequency. It is counted. So instead of doing quadrats, they're doing point sampling along the line instead. So that could be a method used. Okay, let's see what other gadgets they have. So of course, the tree is not the only factor here. Moisture of the ground may change throughout the area. So this here is actually some sort of tool to measure. This is actually light and moisture and pH meter. So all three together, depends on how you flick the switch, and that's pretty cool, right? I thought this tool was pretty cool, and they want to measure those things as well to make sure that, you know, the clover distribution is really affected by the tree and not really the other factors surrounding it. So yeah, they collected those data as well, but of course the main thing is really still looking at the distance from the tree. So how do you make sure that the data that you collected really shows that maybe the distance from tree correlates with the distribution of clover? So maybe the further distance from a tree, maybe the clover gets more. There's more frequency of the clover plants. So you can do that. You can do a stats test, and we're going to learn this in the next lesson that we have together live. We can either use the Spearman's rank correlation, or we can use the Pearson's correlation. And we'll do that in next class, okay? And yeah, so now back to one last slide and one last note on systemic sampling. Now that you get the idea of how it actually measures the distribution of plants along an area, this is one way the information can be actually represented as well, and I think you will see it in a few past years as well. So here is something called a kite diagram. I guess it's called a kite diagram for obvious reasons. It does look like kites flying around, and this actually shows you what plants are available along the rocky shore from high tide area to low tide area. So here's the high tide area and here's the low tide area. Okay, actually I think I might have swapped that around. Never mind. Anyway, the point is you can see that black lichen is more frequent here. Okay, you have different keys here to show you what is what. You can see that it's knotted rag is in the middle here. There's none in the high tide area, none in the low tide area here. And so on and so forth. Like, you can see how it changes, how different weeds, how different rags. Rag is some kind of seaweed-like thing, okay, and how that changes. Different species will prefer different areas along the rocky shore. So yeah, this is distribution. And that's method 2, systematic sampling along line transects or belt transects. So let's go to method number 3, which is a mark, release, recapture. This method is not for small plants. It is not for small non-mobile animals. It is more for stuff that moves around. So the idea is you first capture using a trap, with food as bait maybe, okay, and then you mark those animals. So in terms of snails, you can paint them, clip fur. Actually, snails is quite versatile. You can use quadrat to count them, or you can use mark, release, recapture. In this case, mark, release, recapture. Okay, you can see that this tip here is painted and there's a number labeled there. You can clip a bit of fur off a cat to mark them. Honestly, a lot of neutering programs of cats actually clip the ears of the cat a little bit to show that this stray cat has been neutered in the wild, especially in the UK or the US. So that could be a thing, but make sure you paint it or the fleur clipping doesn't have adverse effects, okay, and make them more visible to predators. You just want it to be quite subtle. We can see it, but it's not that obvious. Now, other forms of marking could be like this tag on bird's claws. This is quite common sight to see, especially in the UK. A lot of geese, a lot of swans have this tag, and that's how they keep track of the animals, okay? So yeah, that's marking the animals. And this is called the mark, release, recapture method. So then you release those animals that you have just marked into the habitat, and you allow some time. You want the animals to mix with the population, but not so long until they move out of the area. So maybe a few days, not too long, right? And then you recapture the animals a second time, not the same mark ones, just a random bunch of whatever you can capture. Okay, and then you calculate the number of marked individuals in that particular second capture, and then you use this index. It's called Peterson index or Lincoln index. Okay, these are actually two people who actually figured this out, and use this to estimate population size. And the equation is this: number marked time 1, so the first time, times the number you captured second time, divided by the number of marked individuals recaptured in the second time. So how do you use this equation? So let's look at our tortoise example here. In day 1, you captured 10 individuals here. So 10, number marked time 1 is 10. Number captured time 2 seems like 10 as well, just so happened. Not always the same, yeah. And you can see the number of marked individuals recaptured time 2 is 1, 2, 3, 4, 5. So that's divided by 5. So if you divide that properly, it's 100 divided by 5, which is 20. So the estimated population size of tortoises in that particular area would be 20. The equation is really based on the assumption that the proportion of marked to unmarked is really the same as the overall population. So the proportion of marked to unmarked individuals in the overall population should be around the same as the ratio here. Okay, if you don't understand that, don't worry. You just gotta use this equation. Okay, you do have to remember this. They don't give you it all the time. I'll give you more practices in the quiz. And yeah, that's pretty much it. This particular one is to again calculate the species abundance of mobile animals, so animals that can move around. So like tortoise, like snails, these are the more mild ones because they don't attack, they're not aggressive, and you can mark them in peace. I'm gonna show you a video right after this to see how they do in snakes and ants, actually very tiny. So yep.
One of the real important approaches we use when either studying snakes or in our monitoring programs with snakes is called mark recapture. And basically what that means is that we capture an animal and we mark it using some type of technique. And then we come back in the future and we recapture that animal. And using that technique, it allows us to do a wide variety of things. It allows us to assess how snake populations are doing over time. It allows us to look at the status of that individual animal in reproductive condition. How often is it breeding? How fast is it growing? Is it eating well? Things of that nature. The primary technique we use to mark snakes is called pit tagging. Each chip has a very unique code, and it's the same type of chip that you would put into your dog, for example, or any other pet for that matter. So we've got a pine snake here. And what I'm going to do is I'm going to restrain it in a tube, and then I'll show you how we go about injecting this pit tag. So there are two places that people usually implant pit tags. Oftentimes people will implant them between the skin and the rib cage on the side of the animal. I typically don't like to do that because I worry about, as the snake moves through the environment, I worry about that catching on things and eventually kind of tearing out of the side. So I typically put them inside of the gut, or what we call the peritoneal cavity of the snake. You don't want it too close to the tail of the snake, which begins here at the cloaca. Usually put it in the lower half of the snake. So the first thing I do is make a loop in the animal's body and hold it tight. And then I take this injector here. You don't want to go down the middle of the snake's body because there's an artery through there that if you hit it, it can really cause a lot of damage. I go in with a pretty sharp angle because I want to break the skin. Then I turn it and flatten it out so the open part of the syringe is pointing against the snake's body. And then just gently inject the pit tag and pull out slowly. Just massage the location to make sure that the pit tag has truly gone inside of the body cavity of the snake, and that animal is marked. So once we have the pit tag inserted into the animal, we put the animal back into the wild, and when we catch it again in the future, we would then use this reader and we would run it along the animal's body, and it would pick up a unique code. We then record the information for this particular snake over long time periods, and we can really be much more effective at monitoring snake populations and ensuring that they're conserved.
for practically all ants, only a proportion of the colony leaves the nest in order to forage, and they bring back the food for everybody else. So one of the measures that one would like to take, one should take, is how many foragers does this colony have? So the real expert in doing these mark recapture studies is actually my former student and colleague Christina Quapich, who is going to give you the details of the method and maybe some other intelligence about that method.
So today we're just going to capture as many workers as we can from the surface, and that will give us an idea of the population that comes out onto the surface each day. And we can follow that population through time with multiple mark recaptures. So what I'm going to begin doing is using the trusted DeWalt vacuum to collect and count foragers as I do it. Now we're going to get a look at our 300 ants. Now we're ready to mark our ants, and we use a fluorescent printer's ink diluted in ether. We use about 20% ink by volume in the ether. So we put our ink and ether into a disposable atomizer, and basically we take our ants and apply 3 or so sprays. And so now they should all be adequately marked, and they'll be marked for the rest of their lifetime. It's been 24 hours after we released the ants that we marked. We come back and collected another 300 ants from the surface of the colony. And now, looking at them under black light, we can see that approximately 50% are marked with fluorescent orange ink. Based on our captures of 300 on day 1, 300 on day 2, and 150 marked ants, we can say that the total forager population size of this nest is approximately 600 ants.
A colony has a fixed amount of labor available to it in the form of the bodies and behaviors of its workers. So how a colony allocates that labor to different kinds of tasks is really important and is clearly a highly selected attribute.
So I hope that was pretty entertaining. I find it entertaining. Anyways, we are pretty much done here. There are 3 things that we learned today. Number 1 is random sampling using frame quadrats. Number 2 is systematic sampling using line, transect, and bulk transect. Number 3 is mark, release, recapture for mobile animals. And that's pretty much it. I'll see you next video. Bye!